Hi, my name is John Quackenbush. I'm a professor of computational biology and bioinformatics in the Department of Biostatistics at the Harvard School of Public Health. And what I wanted to do today is spend a little time telling you about how I got here, what I do, why I do it, why I think it's interesting, why I think it's important. So, unlike many of the people in my department, I took probably a slightly different career path. I actually did a PhD in theoretical physics, and I always thought that I wanted to solve the great problems in the universe. But when I was finishing my graduate degree and starting to do a postdoc, I had a lot of friends who were graduate students and postdocs in biology. And in working with them and talking with them, what I discovered was that in the biological sciences, there was a tremendous opportunity to have an impact, to be able to solve problems, to solve important problems, if you combine a little biological knowledge with a little quantitative skill. And for me, that was really an epiphany. It was a turning point in my career because it let me begin to understand that there were new options and new approaches where we could really make use of new tools, new technologies, new ways of generating data, new ways of analyzing data to address fundamental issues associated with human health. So after working in physics for quite some time and really thinking that that was going to be my career, I applied for a fellowship from the National Institutes of Health to work on something which many of you have probably heard of, but was just getting off the ground. It was something called the Human Genome Project. And what was really amazing about the Genome Project was that it represented a big science approach to looking at the causes of human disease. In genetics, we tend to think about traits being passed from one generation to the next. And we all play the game of genetics when we have children or if we have children in our family. You look at a child and you say, oh, he has mom's ears or dad's eyes or Uncle Bob's hair. And those traits that we observe are traits that are passed from one generation to the next. I mean, that's even the way we talk about them. But the Human Genome Project gave us a great opportunity because rather than looking for individual traits that cause diseases like thalassemia or sickle cell anemia or cardiovascular disease, what we were starting to do was look beyond those individual genes and to start to look for all of the genes in the human genome. And what that was going to represent was an opportunity to really look at big data, to look at the entire collection of blueprints for the individual elements that make up who we are. So the way we think about genomics is that every cell in our body is basically a little machine. It's a little machine made of protein. And the proteins actually have blueprints, which we think of as genes, and those genes are stored in the DNA. And the goal of the Genome Project was actually to get each one of the 25,000 odd blueprints for the 25,000 proteins that make up human cells. And what that was going to do is provide us with a very unique map, a reference map, that we could use using the traditional tools of genetics to start to trace patterns of inheritance in a much more precise way than we could before. And in tracing those patterns of inheritance, to look for proteins that actually had subtle changes in them because of genetic variation, that we could start to relate to a wide range of traits, including traits that cause disease. What that allowed us to begin to think about is to take the kind of genetics we think about doing every day, looking at mom's eyes and dad's hair and, you know, Uncle Tom's smile, and to look at those and really think about the way in which those traits are influenced by the genes that are encoded in the DNA in each of our cells, to combine that kind of information to look at the genes and to begin to look at their functions. So in 1992, I took a fellowship to work on the Human Genome Project. And what we did was we developed the tools and technology to try to take all of the data that we were generating and to take that data and to use it to start to build the blueprint for all those 25,000 proteins, to build a map of where all of those blueprints sat within our DNA. It was a really exciting undertaking because what we were starting to do is to explore the basis of who we are, what we do. And in doing that, there were actually a host of quantitative, statistical, computational problems that we had to solve, wrestling with large quantities of data, taking little snippets of DNA and assembling them together like a jigsaw puzzle in the way that best represented the underlying DNA, but not having a framework in which to put it, not knowing what the DNA looked like. We actually had to use a host of statistical tools to understand where things best fit and to build up the best possible map and to refine that map. We had to use a whole host of quantitative and statistical tools to look through the 3 billion bases that make up the subunits of DNA that are within our cells. Now, 3 billion is a number that's hard to comprehend, right? It's slightly larger than my salary, much smaller than the national debt. But 3 billion is the number of seconds in 95 years. So if you were to be born and started counting one base, one subunit of DNA, every second for your entire life, if you led a very long, productive life and got to 95 years, you would have finished counting through all of those individual bases. When we looked at the DNA in our cells, and we looked at these 3 billion bases, our challenge was to find the genes. How do we actually find the signal, what codes for those proteins, the blueprints for what make up who we are, out of those 3 billion bases? And so again, it was a very rigorous statistical approach to looking for features in the data, combining multiple different types of data, and to try to synthesize it and make predictions that we could then carry back to the laboratory and validate. So the first human genome was sequenced and finished in 2000, announced finished at a big press conference with people from around the world who joined together to bring this data and this project to fruition. What's been even more exciting, though, is now that we have that reference genome sequence, we have the opportunity to start to look beyond just a reference for humanity and to start to look at the individual differences that make us who we are and that drive disease processes. The reason we can do that is actually that there are new technologies that have been developed, new technologies for generating DNA sequence. And these technologies have fundamentally changed the way in which we think about generating data and fundamentally think differently about the way in which we analyze data. The first human genome sequence took, by the time it was fully finished, nearly 15 years and cost upwards of $3 billion. Since 2007, when new technologies were introduced, the cost of sequencing genomes has been dropping dramatically. And in fact, since 2007, the cost of sequencing DNA has dropped by 33% every 3 months. If you look at that trend, starting at the $3 billion genome in 2000 and looking to today, the trend, if you do a little regression and do a prediction, you actually discover that we come to a $1,000 genome sometime late in 2012. We'll probably reach a $100 genome sometime early in 2014. And what that means is the way we think about generating data changes in a very dramatic way. I do cancer research, and a lot of the work we do is to search for mutations that we can associate with the development and progression of cancer. 5 years ago, I would say, well, if I had cancer, I'll mortgage my house and sequence my cancer genome to understand what the mutations are that are driving it so we can best think about treating the disease. Today, the cost has fallen to the point where I can probably put it on my credit card. By 2014, I'm going to be giving my wife her genome for her birthday. And at that stage, our ability to generate data is basically unfettered. We can generate more genomic data today than we have been able to in the entire history of modern biomedical research. And so what we're starting to do is to accumulate large quantities of data. We're starting to tie that to electronic medical records and information about disease. we're starting to link that to the vast universe of published biomedical literature. And we're positioning ourselves to make connections between them, to look at these large populations and to discover trends, to discover genes that are really strongly associated with particular traits, to discover genes whose functions are subverted by mutations that eventually lead to the development and progression of diseases like cancer. To look at those genes in populations and to find patterns that we can use to take what we used to think of as a single disease and to separate them into multiple different diseases, each of which behave differently in the clinic, each of which may respond to different therapies. In breast cancer, we've actually made tremendous progress using this kind of approach. We've looked at the genes, and we've looked at how the genes turn on and off in different patients. As we've done that, we've discovered there are at least 3 major categories of breast cancer. Those 3 major categories are distinguished by the functions that are activated in the individual cells. And as we look at those patients who have breast cancer and who carry these different functional activations, we've been able to group them into different classes and direct therapies to block the processes in those cells, in those patients, that are driving disease. So in one type of breast cancer, what we call luminal breast cancer, cancer expressing something called the estrogen receptor, a little receptor on the surface of the cell that responds to the presence of estrogen. We can now block the activity of that estrogen receptor, now that we've found it, and improve outcome and survival and quality of life for patients who have estrogen receptor positive breast cancer. There's another cell surface receptor called the HER2 receptor, and there's now a drug called Herceptin that we've been able to use to block the activity of that receptor and shut down the processes that make the cells grow out of control. And in disease after disease, we're starting to use these quantitative methods with large genomic data and clinical data to discover new subtypes of disease and to think about new ways of treating disease. So my work now, with the Genome Project mature and our ability to generate technology or generate data due to the new technology is just exploding, my work on a day-to-day basis is to try to synthesize all of this data, to try to synthesize all of the clinical data, to synthesize functional data about genes, and to start to build working models of what happens inside cells. Statistically rigorous, computationally tractable models that we hope will soon allow us to make predictions about what happens in cells when things go well during the normal course of development, and to begin to understand what happens when things go badly when disease develops. So for me, working at the border between quantitative sciences and biological sciences has been a tremendous opportunity. It's allowed me to really start to explore what I think is the great new frontier in biomedical research. It's allowed me to develop approaches that take advantage of big data in ways that we never would have thought we would have access to before, and using approaches that wouldn't have been possible without access to the big data. And in doing so, to really think about how best to improve public health and to improve health and welfare for everyone who suffers, as almost all of us will, from diseases like cancer. So I would really encourage you to think about pursuing a career in quantitative sciences and to think about looking at applications of those quantitative skills that you're building to address these very interesting, very exciting, very challenging, and at the end of the day, really fun to work on questions in modern health, modern biology, modern medicine. So thank you. If we think about why working in this area is such a useful thing to consider, I think for me there are 2 fundamental things that I think about every day. One is, do I enjoy doing what I'm doing? And when I think about myself, I'm basically a big dopey kid who likes to work on hard problems. And I feel extraordinarily fortunate to be working in an area where I can use my quantitative skills together with my knowledge of biology to really do science that's interesting, challenging, and fun. But the other point, and the other important thing for me, is that at the end of the day, I have to make sure I can feed my family and have a roof over my head. And so, working in an area, no matter how exciting it is, where you can't get a job, is not always the best career choice. It may be very rewarding and very exciting, but at the end of the day, most of us have to eat. And so what's been really exciting for me as I've talked to my students and seen them develop their careers is that there are so few people today with good quantitative skills who can address these questions in what's increasingly becoming quantitative biology, that when they finish their degrees, they usually have their choice of jobs. They can pursue a career in academia, go to graduate school, get a postdoctoral fellowship, apply for faculty positions. They get hired into industry, working on cutting-edge projects at the interface between clinical trials, testing drugs, and looking for particular genetic mutations that make people susceptible, responsive to the drugs, or make them resistant to the drugs. They get jobs working in the public sector, using these tools to try to influence public policy by understanding what the data means. So for me, what's been very rewarding is to see all of the students who've worked with me at different stages in their career really move on and have a world of opportunities that are open to them that allow them to take what they've learned and apply it in meaningful ways, and ways that I think many of them are excited about, not only because of the work itself, but because of its potential for improving human health. So if you're a physician, one of the things I think is becoming just increasingly clear to me is that personalized medicine is not just a dream. Personalized medicine is going to be the trend of the future, and it's personalized genomic medicine. As the cost of sequencing genomes continues to fall, I think what we're going to see are increasing applications of this technology to start to try to understand who's susceptible to disease and who isn't. And this is actually a big problem. We tend to think about the different factors that drive development of disease. Some of them are genetic, some of them are environmental, and then there's another factor that we can't really measure called chance. And it's the interplay between genes, environment, and chance that lead to the development of different phenotypes, physical manifestations of traits, including disease. As the cost of generating genomic data falls, what we're seeing even today is genome sequencing, either on a limited scale, looking at individual diseases that we know might be associated with particular traits, or on a large scale, looking for the causes of disease, really proliferating through medicine. Almost every major academic medical center today has a program underway to look at the genetic basis of cancer. And cancer is far ahead of other areas simply because there's been so much more data, because there's been much more ready access to tissue than there has in other diseases. But other diseases are starting to catch up. So we're starting to see approaches using sequencing and using mutation detection and mutation characterization make their way first through cancer and then through almost every other major disease category that has an inherited component. And even in diseases that don't, I don't know if you call them diseases, but in areas in which you don't have a genetic component, say trauma, genomic technologies are being applied to understand processes like wound healing or nerve regeneration. So we're starting to see more and more data on genes, mutations, variational patterns propagate their way into healthcare. Now, you might say, well, that's major academic medical centers. What about the small community hospitals? What about the places where most patients are treated? And the interesting thing that we're seeing, the trend, which is becoming very, very clear, is that as the cost of generating genome sequence falls, there's a greater push to start to use these technologies in the practice of healthcare at almost every level. I've been approached by physicians, physician groups in small hospitals who tell me that they really see a need to take their hospital forward into offering personalized genomic medicine because they want to be able to compete with the major academic medical centers around the world. And the technology now is so inexpensive that they actually see, maybe not it arriving today, but in the next 2 to 3 years, a tremendous opportunity for them to better manage their patient's individual disease by understanding not just the family history, but the genetic history that may have led to the development of those diseases. We're starting to see applications now in newborn screening to try to look not just at a small number of mutations, but a large number of mutations, including rare mutations, that may lead to diseases that weren't economical to test for previously, but are increasingly becoming targets for disease searches. And we're starting to see applications in a wide range of diseases where we're really trying to understand for our individual patients how we can best direct them to therapies that are going to be responsive. So one example is looking in cardiovascular disease and looking at response to a drug called Plavix. And what we can do today is actually screen a host of mutations that have been associated with response to the drug. And for each individual patient, try to predict their likelihood to respond. So I think for physicians, this is really a great opportunity to build your quantitative ability to take the data sets which are going to grow and to be well positioned to make use of them, either to ask research questions or to try to best help your patients in the years to come to make sense of the disease. Now, I've talked a lot about genomics. The other important piece of this is population-based science. And one of the things we know very, very clearly is that when we look at disease, the only way to find the patterns that we can eventually use to find new therapies and to direct individual patients to the appropriate treatment is to look at those large populations and to make sense of the patterns that exist in those patients and in those populations. So if you're a physician, I think you're making a very wise choice in taking this course.